Welcome to the European College of Neuropsychopharmacology podcast, where we interview the most interesting people in translational neuroscience.
Hello, I am Silvia Bruzzone. I am a PhD student at Copenhagen University Hospital, and today I'm glad to welcome Professor Roger Adan at the podcast. Hi, Roger.
Hi.
So Roger is a full professor in molecular pharmacology at Utrecht University. His research focuses on the molecular and neural pathways of feeding behavior, obesity, and eating disorders. And to start with, I would like to ask you something about your scientific career. How did you get into this research topic, and how did you start?
Oh, yeah, that's a long story. So I used to work in what was in those days called the Rudolf Magnus Institute, which was led by people like David de Wit, who worked on neuropeptides, but also Jan van Rey and Willem Hendrik Gispen. And so I started to work there on neuropeptide gene regulation because I came in as a molecular biologist. And in those days, like in the early 90s, molecular biology, in the 80s, the molecular biology was really coming up. So I was supported by Peter Burbach as a supervisor, and I developed those technologies and investigated vasopressin and oxytocin gene regulation. And then I was very interested to do a bit more translational research, and I thought, well, of course, if you want to mimic these systems, you would like to work on receptors. So after finishing my PhD, I've been interested to clone receptors. In those days, you could clone receptors, and of course the genome was not known at all. So, and of course we didn't know what kind of receptor subtypes existed. And the first few neuropeptide receptors were cloned, meaning that indeed these receptors existed. It was those days. And there was also a lot of work on melanocortins and HTH and MSH fragments in those days because they had behavioral effects. But it was difficult to convince people that these effects were real. So we really like to figure out the cloned receptors for that. But rather than going abroad, which was my plan, they offered me a job to stay as a postdoc at the same institute. So I thought, well, if I just can stay in the same house and do the things I usually do and biking to lab, why not? So I kept on doing this, and I started to clone receptors, and we also got receptor clones from collaborators. We started to express them on cell lines. So for years, I worked only in vitro, figuring out what kind of peptides that we tested before, because we tested all kind of truncated peptides and so on, and we had all kind of pharmacological profiles of behavioral effects of these peptides. And then, of course, with the cloning of receptors, we could figure out which of these fragments of peptides were binding to those receptors. So that kind of work I did. And it rapidly moved in cloning melanocortin receptors. And there are 5 subtypes. And so you have the MSH receptor in the skin, which is the MC1 receptor. On the adrenal gland, you have the MC2. And then there's 3 receptors which are expressed in the brain, MC3, MC4, and MC5. And by the time we were working on this, it was discovered that some of these were important for regulation of energy balance. And in those days, I didn't study energy balance at all. But it turned out that there was a genetic defect in a mouse model that overexpressed an endogenous antagonist called agouti, which blocked melanocortin 1 signaling in the skin. And that's explained that this mouse had a white skin. But at the same time, the mouse was very obese. And the idea was, of course, if this is in the periphery, in the skin, acting on a receptor, the MSH receptor, MC1, then, of course, we know this receptor is not expressed in the brain. Maybe there's another melanocortin receptor which is expressed in the brain. So then when we got clones of these receptors and we started to investigate them, and we discovered also that indeed these endogenous antagonists, and in the meantime there were more, there was not only agouti, but there's also a brain version of it, agouti-related protein, they acted as inverse agonists. So they didn't only block the approach of the agonist to the receptor, but at the same time they reduced the constitutive activity. And there was then a paper which we made, which was showing that HRP was an inverse agonist of the MC4 receptor. There was a paper in the Molecular Endocrinology, in those days, high impact journal, together with my postdoc of those days, Martin Kass, who's now ECNP president, and he invented a title of a trans paper called Inverse Agonism Gains Weight. I thought that was very cleverly thought of. So, because if you block the melanocortin-4 receptors or you delete the receptors from a mouse, then you get a very obese phenotype. So, and I was studying, of course, everything in vitro. But then I realized that I was competing with pharmaceutical industry, and they told me, well, you really, if you want to develop a drug, it really needs to be a small molecular weight compound. So, and we could generate our own peptidergic ligands, which we also stabilized and so on, and we had patents and so on, but the industry was not at all interested. If we now jump 20 years later, industry is very much interested in peptides because nowadays it's very normal to put them in injectables. So if I would have kept in that line, I may have developed even more potent ligands, and maybe the patents would really have been sold now to pharmaceutical industries, and I would be rich. But that didn't happen.
How unlucky.
I made a jump because I thought, well, the real thing what we can do as an academic institute is to figure out how this works. So how is it that in the brain, if you block MC4, that you increase your appetite? And the thing
So you
was
stayed in academia because you wanted to understand more about the mechanisms.
Yeah, I wanted to understand the mechanism, how it works. But then at the same time, so we were closely associated with people that then still worked at the psychiatry department of the hospital. And there were also anorexia nervosa patients. And the idea came that we thought, well, if there's an endogenous antagonist, HRP, that normally blocks this receptor, and that in fact if you inject it you get hunger and you start to eat, maybe there's a mutation in this particular gene in anorexia nervosa patients which explains that they don't eat and that they don't feel the hunger. So we started a collaboration with the Eating Disorder Clinic and gathered DNA samples, collaborated with Johannes Hebermann in Germany, who had also had samples. And in this way, we found an association between a variant in the HRP gene and a susceptibility to develop anorexia nervosa.
Oh, interesting. So you managed to translate your results from preclinical studies to clinical.
The thing a bit now with the genome association studies, this is not replicated. So maybe the sample size needs to be increased to find it again. But so far, it's not a very strong finding. But it was the starting point for a whole... for me, moving into a completely different field of in vivo pharmacology. So we started to investigate how ligands that were specific to these receptors impacted on the brain. And it also was the seed for my interest to collaborate with clinicians. So I still have a very small position at an eating disorder clinic, and we have bi-monthly meetings with them to discuss research and nowadays also engage patients and so on.
How do you model anorexia nervosa in animals? Because it sounds more like a uniquely human sort of disorder.
Yes, of course, any animal model has its limitations. But in this case, this animal model that exists is that you expose animals to cages with a running wheel or a running disc. And of course, rats and mice really love to run in the wheel or on the disc. But something very strange happens if you start to food restrict them and give them access to food for 1 to 3 hours a day. What would be clever is that they would stop running to save energy, but they don't. They increase their running, in particular in the light phase when they usually are resting. So therefore, by the combination of food restriction with running in the wheel, they lose a lot of weight. So their neuroendocrinological profiles are very similar to anorexia patients, and I'm sure that the changes at the level of the hypothalamus and the rest of the brain are also the same. So that is the part we mimic, but of course we cannot mimic the body image disturbance. And if we give food back, then of course they start to eat. So therefore now we also have a version of the model where we first double conditioned taste aversion. So we give the mice chocolate to eat, and then after this experience, we inject them with lithium chloride, which gives you a nausea feeling. If you then expose them expose them to this activity-based anorexia model, and then give them chocolate, they won't eat it anymore. So this mimics the conflict that anorexia nervosa patients have, like if they are confronted with food, because in this model, we attributed negative value in a cognitive manner to that food because we don't inject them a second time with lithium chloride, so they don't get ill of it. They can just eat the chocolate, but they don't because they remember this made me ill, so this is not good for me. So we're now mapping the brain pathways that are involved in this.
How interesting. So do you have any hypotheses about what are the brain areas that might be most involved?
Yeah, we're currently mapping those brain areas, but definitely we think there's an involvement of the dopamine system, the midbrain dopamine system. 10 years ago, we discovered that if we injected leptin into the VTA, we can suppress this hyperactivity that develops in the model. And interestingly, levels of leptin are also associated with hyperactivity in patients. And to come back on the guy that introduced you, Johannes Hebebrand in Germany, he started treating his patients with metroleptin, which is an analog of leptin. And it has surprising results. These patients apparently open up for therapy and they start to eat. And that's really intriguing because also, in the animal model, we see that they stop their running behavior. So we think that this has to do with an effect of leptin on suppressing the hyperactivity that develops in the model on in the dopamine system.
Amazing. That's super interesting. We've talked about mechanisms and instead about more epidemiological features of eating disorders. Eating disorders have been steadily increasing in the world. Do you have any idea of why this is the case?
Yeah, of course, there's always, besides genes, also environmental interactions. So it's well known that stressful events and other traits like perfectionism and anxiety contribute to the development of anorexia nervosa. So there's a clear link between hyperactivity and anxiety in anorexia nervosa patients. And we also see that exposure to this model, there's also a relationship between anxiety, anxious behavior and hyperactivity in the animals. So it looks like they are starting to run in order to overcome the anxiety, something like that.
Okay. Is there any current medication for anorexia nervosa that is targeting specifically the anxiety, and does this help alleviate the symptoms?
There's currently no specific treatment for anorexia nervosa. Yeah. There's no biological treatment, like I should say. So the treatment consists of trying to encourage patients to eat, to treat them in a psychological manner. In my view, having any disease is a balance between disease load and kind of resilience, how you deal with it. And the resilience is something that is treated in psychotherapy. And I think the cause of disease, that is, of course, more biological. And so we first have to unravel the biological mechanism underlying the disease before we can think of development of biology-based therapies. And that's what we are doing.
Of course, yeah. So you work on anorexia nervosa, but you also work on obesity, which is sort of the flip coin.
Yeah. So because it's the same kind of systems play a role in it. So my colleague and old PhD student Frank Meijer is studying stress-induced eating and binging. So that's the other side of the coin. But we are working with similar technologies, using optogenetics and chemogenetics and in vivo electrophysiology and electrovisualized electrophysiology to map these circuits, record for them, manipulate the activity of these circuits. Because it started with manipulating the brain with drugs, and then we were able to modify genes. But then with the occurrence, the possibility to use chemogenetics and optogenetics, we can now manipulate whole cells. And we can also record from whole cells using, for instance, fiber photometry, which we do quite a bit. So we want to know, so the strategy of our research is that we map the circuits that are involved, we record when they are active related to the model, and then we try to manipulate them in order to see whether manipulation could have a therapeutic effect. And then we would find, using single-cell RNA sequencing and transcriptomics, we'd like to identify druggable genes in those cells that are engaged upon exposure to the model.
Interesting. Have you found in your research any targetable gene for possible therapies also for obesity?
I mentioned, of course, leptin. That's the first one. But of course, ghrelin could be one as well. So I also have a guest professor position at the University of Gothenburg, where I collaborate with Suzanne Dixon. And of course, she's a ghrelin expert. And the idea there is perhaps to to develop intranasal administration of ghrelin receptor agonists further as a kind of add-on treatment to anorexia nervosa. And of course, we expect to find targetable genes the coming years that we will target.
Well, very curious to see where that goes. Okay, well, you've had a very interesting research career and you mentioned a lot of very intriguing findings. But before finishing, I would like to ask you one last question, which is there any advice that you would give to early career scientists for the development of their career in academia?
Networking is really important. And I did not start networking early enough in my career. I only discovered that networking was really important maybe the last 10 years or something. I was just writing grants and I got these grants and went on, but nowadays you need to work together in research, and you do that by building networking. And in particular, I think ECNP is very helpful there because they invest in network building a lot. So ECNP is an important social community for me, and I'm also a member of 2 other societies. And this is really something that you should pick up early in your career. Being part of a society is going to help you.
That's certainly a great advice. Okay, well, thank you so much for being with us today.
Yep.
Bye.
You're welcome.
Thanks for listening to this podcast from the European College of Neuropsychopharmacology. The views and opinions expressed in this podcast are those of the interviewee and not necessarily the official position of the ECNP. And of course, this should not be the basis for clinical treatment decisions.